So today's class discussion is a continuation of our last class discussion on the protein kinase structure. So we discussed in the last class, and I hope you have gone through and you followed the discussion by going through the textbook. So there are two lobes of protein kinase, and there are different substructures which have got very important role. And there is a small subunit, and there is a larger subunit. This N-terminal end, and there is a C-terminal end. So the conformations are very important that are related with its functional, whether activated state or inactivated state. So inactivated state and activated state, mainly the difference is the exposure of the active site. So active site is the region where phosphorylation of the substrate is taking place. So for that function, what has to be done by this enzyme? So it has to be bound with an ATP, then it has to get exposed its active center so that the substrate can bind to the active center where phosphorylation is being done, right? So those conformational changes are mainly like closed conformation to open conformation, and closed is inactive because the active site is inaccessible, whereas the open conformation is a conformation in the active state of the this kinases where the active site is occupied by the substrate, and the small lobe is attached with the ATP, and the gamma phosphate is transferred to the substrate at a particular site. Last class we have discussed protein kinases have been divided into four major classes based on the site at which this particular kinase is responsible for phosphorylation in the substrate, right? So, like the first one was serine threonine kinases, which will phosphorylate at serine or threonine hydroxyl groups, and then tyrosine. Next is tyrosine kinases. So these three kinases are responsible for phosphorylation at hydroxyl residue. And other kinases can be at carboxylic acid residues or at the amide residues. So we had already discussed about the important structural elements. So the protein kinase has got important structural elements such as P loop, C helix, catalytic loop, activation segment with activation loop, and auto-inhibitory elements. So these are the important structural elements that are responsible for the function of the protein kinase. And as we discussed, ATP binding to the small subunit is the one which donates the phosphate group, and here alpha, beta, gamma. So the terminal phosphate is the gamma phosphate, which is being transferred to the substrate, right? So the first one, P loop, is also called as glycine-rich loop. This already we had discussed, and this is the N-terminal lobe, which is responsible for binding to ATP. Okay, so in the N-terminal smaller lobe, ATP binding loop is nothing but the P loop, or this is also called as glycine-rich loop, because this is rich in its consonant sequence, the glycine. So glycine, X, glycine, 2X, and G. So these three glycine, and there are three non-specific amino acids in the sequence. So this conserved sequence is the glycine-rich sequence present in the smaller loop and is responsible for the binding, anchoring the non-transferable ATP beta. Here there's a mistake. Alpha beta non-transferable. This will be within the small lobe of the protein kinase enzyme, whereas the gamma phosphate is known as the leaving group. Okay, you have corrected, right? This I told in the last class also. Alpha beta phosphate is the one which is retained in the smaller subunit, whereas the gamma phosphate is the phosphate group which is transferred from ATP to the substrate kinase. Okay, so that is mainly done because of the binding of ATP in the small subunit, that's called P loop. So by binding and positioning ATP appropriately for gamma phosphate transfer to the substrate, the P loop control nucleotide affinity, specificity, and gamma phosphoryl transfer. Okay, so these are the main important functions of the small subunit by which ATP, the donor, will be bound because of the affinity and specificity. It can retain alpha beta phosphate ADP within the small subunit, whereas gamma phosphoryl transfer is made possible, and these all functions are because of P loop. Understood? Next, we discussed another one is C helix. This is uh, in the internal lobe again. So this is the C helix, and this is the P loop here, which we discuss now. So in many kinases, the residue E91 of the C helix form salt bridge to an invariant residue K72 within the N-terminal. So these are important, E91 and K72. So what is the E91? E mentions which amino acid. K means which amino acids. This you should know. So these two amino acids at these particular positions in the C helix are very important in forming the salt bridge, and that positions the ATP phosphates. So regulatory mechanism is again plus and minus, whether the kinase activity needed or not. So those functions are responsible for modulating the kinase activity by altering the conformation of the C helix. So C helix is mainly having regulatory functions by which it can control the activity of the protein kinase. That is the function of the C helix. Understood? So next one is catalytic loop. So catalytic loop is here that you can see. And this catalytic loop is located basically at the base of the active site. This is active site catalytic loop. And active site is the site where substrate can bind. So little bit at the base of this active site, you have the catalytic site. And this contains a conserved residue D166. So this is very important. In the next topic, we can discuss in detail about this D166. We are going to discuss how this protein kinase performs the reaction, how it transfers, what are the different residues within the kinase responsible for transferring gamma phosphate, and what other moieties are having role in the protein kinase reaction. Okay. So D166 is the, what is D? Can anybody tell? Single letter D, which amino acid? So earlier E we discussed, K discussed, now it is D. Can anybody tell what is D?
So aspartic acid.
Very good. Okay, what is E? Which one is E letter?
Sir, is it glutamic acid?
Okay. So all of you should know what is the first letter and which amino acid, because here we are positioning the importance of these kinases with respect to the particular amino acid of that particular position. So here we are discussing in the catalytic loop of the protein kinase D166. D is for aspartic acid. So at that particular position, aspartic acid 166 has got very important role, and this presumed to be having role in catalysis. What is the catalysis function here? Transfer of gamma phosphate from ATP to the substrate. So furthermore, a conserved another one N171 residue is also having a role in this catalytic. So first important one is D166 and then N171. So all of you should know what is N, which amino acid it is. Next one is activation segment and activation loop. Activation segment and activation loop, or T loop. T loop for cyclin dependent protein kinase. So this is another important structure. So activation segment is the region between and including two conserved tripeptide motifs. Okay, so this is tripeptide motifs, two tripeptide motifs, DFG and APA. This is our motif. So within the larger lobe, this is the larger lobe, right? So within this particular moieties are present, and this about 20 to 35 amino acids consists of. So the conserved tripeptide motif is about 20 to 35 total in amino acid length that forms this T loop. So the other activation segment include Mg2 plus binding loop. Mg2 plus again another important cofactor for the protein kinase function that also will come across in the next topic of our discussion, protein kinase reaction. So Mg2 plus binding loop is there within the activation loop, and this comprises the DFG motif. Activation loop, P plus 1 loop is there, and that is involved in peptide substrate binding. So these are the functions you need to by heart, and you have to write it in your answers. Okay. So these are the different regions within the activation loop or T loop, and these are basically about 20 to 35 amino acid containing tripeptide motif, conserved tripeptide motifs, is known as DFG-APE motif, and also it has got another motif that is Mg2 plus binding loop, and this is nothing but again containing DFG motif, and activation loop uh, has P plus 1 loop, which is involved in peptide substrate binding. So here the substrate is the another kinase which is being activated. Okay. So the residue D184 of the conserved DFG motif is involved in metal ATP binding. So in the protein kinase reaction, when we discuss all these things in detail, we are going to discuss. So D184, earlier it was D166, right? In the small lobe, D166 is very important. But here it is D184 in the larger lobe, and that is containing DFG motif. It is responsible for metal ATP binding. This metal is in a way it holds this ATP within the protein kinase. Okay, so that is because of this D184 present in the larger subunit activation loop, and serine 309 or tyrosine present in the activation loop can undergo phosphorylation and attain active conformation of the kinases. So we discussed that in signal transduction, these protein kinases, if you see map kinases, all these are nothing but kinases. They undergo kind of after activation, they undergo phosphorylation and then become active. Then activated again acts on another protein kinase of a similar structure. So these protein kinases basically they are nothing but again they have the moieties within the activation loop, serine 39 or tyrosine, and they undergo phosphorylation. When they undergo phosphorylation, they become active structures. Active structures mean they open the structure from the closed structure, they undergo transition, and the open structures will then provide site for the substrate binding, right? So in many inactive states of protein kinases, the activation loop collapses as just we discussed. This conformation basically inactive state means the activation loop collapses. Collapses means it is not accessible. It is hidden, buried within the structure. Collapses into the active site. So what happens because of that? It blocks the binding of both nucleotide and peptide substrate. So peptide substrate is the one here which is getting phosphorylated. So both nucleotide, ATP and peptide substrate that they cannot have the place for binding and the reaction to go on. So upon phosphorylation from the inactive state, it moves away, the closed collapsed structure moves away from the active site and attains an open conformation, allowing substrate binding and catalysis. That's what we discussed. So this changes in the active site in such a way that phosphorylation. So why phosphorylation is inducing the transition? Because protein is a charged molecule and its conformation is mainly stabilized by its native charge. So native charge is very important for all these proteins, and the charge is again, it depends on how much, whether it's acidic protein, basic protein, neutral protein. Acidic protein means if it is rich in acidic amino acids, so negatively charged, net negative charge protein, net positive charge protein, or neutral protein. Depending on type of amino acid that it contains, its conformation also depends on. Now, anything that responsible for change in its charge, nothing but can alter the conformational change, and in a way, it impacts the functionality of that particular protein. So same way here, basically the phosphorylation mechanisms of the substrate protein is responsible for open structure formation. Understood? So that way it provides the site for binding substrate. So critical for this conformational change, as I told, electrostatic interactions, because protein is basically charged molecules, and this electrostatic interactions status will be altered upon phosphorylation, because phosphate has got negative charge, and between one of the phosphate residue of the loop and basic pocket containing arginine aspartic acid motif, while conserved among kinases regulated by phosphorylation. So this particular motif is undergoing the impact of this phosphorylation, okay, and that leads to open conformation of the kinase, nothing but it is active state. So the position and number of phosphorylation sites, which active site is undergoing phosphorylation and how many, that is variable among different kinases. Understood this point? So this all comprises about the activation segment or activation loop or T loop for cyclic dependent protein kinase, which is the very important site for the activity of these protein kinases. All of you understood some points you need to practice and keep memorized. So the presence of phosphate residues create a network of interactions that properly orient C helix and the catalytic residues and promote lobe closure. C helix, we discussed that it is the one which is having regulatory function, right? So during the after phosphorylation, this is forming the network of interactions, and these will influence the C helix and then catalytic residues, promoting the lobe closure. So the lobe has to here catalytic residues and lobe closure. It should not be lobe closure. Please check this. This is open structure, right? When presence of phosphate residues, that is after phosphorylation, this is creating network of electrostatic interactions like in the other point, and this properly orients C helix. All these changes are making this protein kinase to become active state. Active state is the open structure, it's not closure structure. So this you need to again check. So for certain kinases like MAP kinases, in textbook also sometimes you can find wrong things, okay? So you have to be attentive. So for certain kinases like MAP kinase, I told that it is microtubule associated protein kinase. That was earlier nomenclature, but now it is renamed as mitogen activated protein kinase. So phosphorylation of the activation loop facilitate homodimerization of the kinase that is required for the nuclear localization of the enzyme. Homodimerization means two of the molecules after phosphorylation they become dimers. So two kinases will become dimer, and then they are responsible for nuclear localization. That is the step that you will see in case of what is the step extra for MAP kinase? Homodimerization. Okay, that is specific for MAP kinase. Next one is auto-inhibitory sequence. So along with this activation mechanisms, Also it has got auto-inhibitory sequences within the structure, and these have got, as the name suggests, they are responsible for negative control of this protein kinase function. So these sequences help to fix an inactive conformation by intramolecular binding to the substrate binding site. So these are the sequences that help fixing an inactive conformation. So making active conformation to inactive conformation fixing by intramolecular binding to the substrate binding site. So substrate binding site is nothing but activation site. That has to get changed so that it has become closer structure. So these elements may fold into an active site, blocking the binding site of both the nucleotide and peptide substrate. Okay. So in the active site, when they become folded structure, they block. So active site, when they become folded, that is closed structure, it blocks the binding of both nucleotide and peptide substrate. As these sequences do not have phosphorylatable residues, inhibitory sequences. These elements are also known as pseudo-substrates. So autoinhibitory sequence elements of protein kinases are known as pseudo-substrate because they don't have phosphate groups, but they are responsible for the closer structure and helps in activation, active site to fold by undergoing intramolecular binding to the substrate binding site. So this way, these are called pseudo-substrate instead of phosphorylation. Phosphorylation we discussed in case of activation loop, but in case of inhibitory sequence, it is because of the intramolecular binding with the substrate binding site, not because it is initiated with the phosphorylation. Understood? So that's why it is called pseudo-substrate with the autoinhibitory. Negative regulation of the protein kinase is known as pseudo-substrate. So this is what we have discussed now. This is the C helix, which is having the regulatory role, glycine loop or P loop, as we discussed. And this is in the smaller lobe of the protein kinase, where ATP binding is taking place. And then this is the activation loop. Then activation loop is with the C helix, and then activation loop, and then the last one is inhibitory sequences. Okay, so all these regions are having very important role to perform protein kinase function and its regulation. So any questions you have? That is all for today's discussion. So if you have any questions, let me know. So some of the things you need to memorize, so keep practicing. And all this, you can use it as a notes directly because this all material is from your Gerhal Cross book, and it is good to go ahead for making answers for these questions. This question is very important. You can expect for a big answer question. Protein kinase structural elements. Any questions? Any questions? Have you followed all of you? Please unmute everyone and tell me.
Yes, sir.
So if you have followed, then
Yes, sir.
and no questions from you for the discussion, then I'm going to stop here today, and tomorrow we are going to continue with our next part of discussion. So tomorrow maybe I will take molecular biology. Okay then, see you all.